Dear viewers, greetings. In this video, we are going to see about field applications of bioinoculants to agricultural crops. 6 methods of field applications of bioinoculants were regularly practiced in the present agriculture. They are seed treatment, seedling root dip method, soil application method, furrow application, drip irrigation, and foliar spray. The first method of field application is seed treatment. Seed treatment is the most common and efficient method, ensuring the microbes are in direct contact with the germinating seed. Rhizobium species, Azospirillum species, Acetobacter species, and Phosphobacteria are applied as seed treatment. For seed inoculation, apply solid inoculant at 200 grams per acre or liquid inoculant at 50 ml per acre. Procedure for seed treatment. 200 grams of the inoculant is mixed with 200 ml of rice gruel or jaggery solution to make a slurry. The seeds required for an acre are mixed in the slurry so as to have a uniform coating of the inoculant over the seeds and then shade dried for 30 minutes. The shade dried seeds should be sown within 24 hours and one package of the inoculant is sufficient to treat 10 kg of seeds. Crop recommendation for seed treatment. For legumes, seed treatment with Rhizobium species helps to improve nitrogen fixation and nodulation. For cereals, the use of Azospirillum, Acetobacter or phosphate solubilizing bacteria is very helpful for seed treatment which enhance the root growth, nutrient uptake and plant yield. For oilseeds like soybean and sunflower, seed treatment with Rhizobium or phosphate solubilizing bacteria helps for better nitrogen and phosphorus availability. For vegetables, the use of Trichoderma species, Pseudomonas fluorescens or Bacillus species prevents seedling diseases and improve early growth. Advantages of seed treatment. The seed treatment is efficient, low cost, target the seed directly, protects seeds from pathogens and pests, improves germination and uniform seeding growth, reduces the need for soil applied chemicals, and finally, seed treatment can include beneficial microorganisms to enhance nutrient uptake. Limitations of seed treatment. The first limitation is seed treatment can be messy. The second limitation is improper drying can damage seeds. The third limitation is microbial exposure to seed treatment chemical is a risk. The second method of field application is seedling root dip method, which is also called as dip seedling method. Seedling root dip method is used for transplanted crops like tomatoes, peppers, cabbage, rice and trees. Azospirillum species is used for seedling root dip, particularly for rice. For seed root dip inoculation, apply solid inoculant at 400 grams per acre or liquid inoculant at 150 ml per acre. Procedure for seed root dip method. 400 grams of inoculants are required for 1 acre and mixed with 40 liters of water. The root proportion of the seedlings is dipped in the solution for 5 to 10 minutes and then transplanted. Crop recommendation for seed root dip method. For rice, dip seedlings with Trichoderma species, Pseudomonas fluorescens or Rhizobium species to prevent seedling rot and root diseases and also for enhancing nitrogen fixation and promote early growth. For vegetables, dip roots with Trichoderma species or PGPR solution for reducing damping off and improve root establishment. For cotton, treat seedlings with beneficial microbial suspension to protect the crop against soil-borne pathogens and enhance root vigor. For sugarcane, dip seedlings with biofertilizers like Azospirillum or Trichoderma species to improve their survival and early growth. The advantage of seed root dip method includes excellent root colonization, highly effective for transplants, protection against soil-borne pathogens, improved root establishment, enhanced nutrient uptake, and increased seedling survival. Limitations of seed root dip method. The first limitation is labor intensive, and the second limitation is not suitable for large scale direct seeded crops. The third method of field application is soil application method or direct broadcasting method. Soil application or direct broadcasting method directly introduces microbes into the soil. Procedure: 4 packets containing 800 gram of inoculants are mixed along with 25 kg of soil or farm manure or compost fertilizer and are kept for overnight. Next day, the mixture is incorporated in the soil at the time of sowing or planting. Crop recommendation for soil application method. For cereals like rice, wheat and maize, Azospirillum, phosphate solubilizing bacteria or Acidobacter were applied along with the basal fertilizer to improve nitrogen fixation and phosphorus availability. For pulses like chickpea, pigeon pea and lentils, Rhizobium inoculants were broadcasted along with phosphorus fertilizer to enhance notilation and nitrogen fixation. For oilseeds like groundnut, sunflower and soybean, Rhizobium, phosphate solubilizing bacteria or Azospirillum were applied to improve nutrient uptake and growth. For vegetables like tomato, cabbage and chili, microbial inoculants were used in the soil before transplanting to improve soil fertility and seedling establishment. Trichoderma species, Pseudomonas fluorescens or mycorrhizal fungi were applied in the soil to suppress soil-borne pathogens and enhance root development. Advantages of soil application method. The advantages of soil application method includes it is good for established perennial crops. It is less harmful to microbes than seed treatment with pesticides. Uniform nutrient distribution. It improves soil fertility. It is very simple to apply on a large scale without specialized equipment. It supports early crop growth and provides nutrient at the time of sowing and promoting healthy seedling establishment. Soil application method is suitable for cereals, pulses, oil seeds and vegetable crops sown by broadcasting or in rows. And finally, it reduces nutrient wastages. Limitations. There are two limitations for soil application method. The first one is the soil application method requires a high quantity of inoculant, and the second limitation is it is less targeted than seed treatment. The fourth method of field application is furrow application. Furrow application is a variation of soil application where the inoculant suspension or granules are delivered directly into the seed furrow during planting. Furrow irrigation is a surface irrigation method in which water is applied to crops through small parallel channels called furrows made between crop rows. Water flows along the furrows by gravity and infiltrates into the soil and providing moisture to plant roots. Crop recommendation for furrow application. For cereals like wheat, rice, maize and sorghum. Azospirillum species, Pseudomonas fluorescens or phosphate solubilizing bacteria were applied in furrows to enhance root growth, nutrient uptake and yield. For pulses like chickpea, pea, lentils and green gram, Rhizobium species and phosphate solubilizing bacteria were applied in furrows to improve nitrogen fixation and phosphorus availability. For vegetables like tomato, brinjal, cabbage and chili, Trichoderma species, PGPR or phosphate solubilizing bacteria were applied through furrows to protect seedlings from soil-borne pathogens and promote healthy growth. For sugarcane, furrow application of Trichoderma species or Azospirillum species enhances root establishment and early growth. Advantages of furrow application. The advantages of furrow application includes precise placement near the seed, efficient use of product, efficient water use, suitable for row crops, reduces weed growth, easy to construct and manage without advanced equipment, allows controlled water flow, reducing the risk of water stagnation in field, and finally improves nutrient uptake. There is only one limitation for this furrow application. It is the furrow application requires specialized planting equipments. The fifth method of field application is drip irrigation. In drip irrigation, the liquid inoculants can be injected into irrigation system. Drip irrigation is considered as one of the most efficient irrigation technique because it minimizes water loss due to evaporation, runoff and deep percolation. The deep irrigation is an advanced method of watering crops in which water is delivered directly to the root zone of plants, drop by drop or sprinkling through a system of pipes, valves, emitters and tubings. Top recommendation for drip irrigation. For vegetables like tomato, chili, brinjal and cucumber, Trichoderma species, Pseudomonas fluorescens or Azospirillum species were applied through drip irrigation to protect roots from soil-borne pathogens and enhance growth. For fruit crops like banana, papaya, guava and citrus, Acetobacter species, Azospirillum species or mycorrhizal fungi were used via drip irrigation system to improve nutrient uptake, fruit quality and fruit yield. For sugarcane and cotton, bioinoculants were applied through drip irrigation to promote root development, soil microbial activity and overall productivity. Advantages of drip irrigation. The drip irrigation directly reaches the plant root for better colonization. The drip irrigation improves nutrient uptake like nitrogen, phosphorus and potassium and plant growth. It reduces the amount of inoculant needed and the drip irrigation increases crop yield and root development. Drip irrigation helps to control the soil-borne diseases. The drip irrigation enhances water use efficiency and it is eco-friendly and reduces chemical fertilizer use and it can be combined with fertilizer in fertigation. Limitations of drip irrigation. There are two limitations of drip irrigation. The first limitation is that drip irrigation requires compatible irrigation infrastructure. And the second limitation of drip irrigation is water pH and chlorine content can kill microbes, so it may need filters or dechlorinators. The sixth method of field application is foliar spray. Foliar spray is a method of applying nutrients, growth regulators or pesticides directly onto the leaves of plants in the form of a fine mist or spray. In foliar spray, the nutrients are absorbed through the stomata and epidermis of the leaves, allowing quick correction of deficiencies and rapid response in plant growth. The use of foliar spray is less common but used for certain phyllosphere microbes or biocontrol agents. Foliar spray is the best for applying biocontrol agents like trichoderma or pseudomonas for foliar disease. In foliar spray, the inoculant is mixed with water and sprayed directly onto plant leaves. And adding a surfactant can improve the adhesion ability. Crop recommendation for foliar spray. For cereals like rice, wheat and maize, azospirillum and Pseudomonas fluorescens improves the growth, nutrient uptake and disease resistance. For legumes like soybean, chickpea and groundnut, rhizobium and trichoderma enhances the nodulation and protects against foliar diseases. For vegetables like tomato, chili, cabbage and okra, Bacillus subtilis and Pseudomonas fluorescens controls the leaf blight, powdery mildew and boost fruit yield. For fruits like banana, mango, citrus and grapes, Trichoderma and Pseudomonas fluorescens improves disease resistance and fruit quality. For cash crops like sugarcane, cotton and tea, Acetobacter and Trichoderma species promotes the growth and reduces foliar infections. Advantages of foliar spray. The advantages of foliar spray includes rapid correction of micronutrient deficiencies, for example, zinc, iron, boron and manganese, saves cost by reducing wastages of fertilizers, useful for crops with shallow root system, delivers water directly to the plants, to the root zone and minimizing evaporation and runoff, enhanced nutrient uptake, maintains optimal soil moisture for consistent growth and productivity, reduces weed growth, suitable for uneven, sloping or arid lands, saves labor and energy because it can be automated using timers and sensors for precise irrigation, and finally reduces water wastage and minimizes leaching of nutrients into the soil. Limitations of foliar spray. There are three limitations. The first limitation is it provides temporary effect, not a substitute for soil fertilization. The second limitation is excess concentration of foliar spray may cause leaf scorching or burning. The third and final limitation is the weather conditions like wind, rain and high temperatures affects the effectiveness of the foliar spray. Dear viewers, that's all about the field application of microbial inoculants. Thank you for your support. Thank you.